Today, I'm not going to talk about an actual robotic system, but about the modeling of a biological system whose understanding can potentially inspire autonomous robotic swimming devices in viscous fluids in both microscopic and macroscopic environments, which are self-propelled. So as many of you will know, swimming at the micron scale is very different from swimming at the macroscopic scale, and this difference is codified in the dimensionless Reynolds number, which is a measure of the ratio between inertial and viscous forces. And the Reynolds number of swimming microorganisms is typically very small due to their speeds and dimensions. And so in such viscosity-dominated conditions, in order to swim, it is necessary to break both a spatial and a temporal symmetry. And one of the most common universally used strategies is the one adopted among many by the model organism Chlamydomonas reinartii. So this is a unicellular biflagellated alga, 10 microns in diameter, which swims thanks to two whip-like appendages called cilia or flagella, which beat at a frequency of 60 hertz and allow the organism to swim at 100 microns per second on average. The beat of each flagellum, as you can see, is characterized by a power stroke and a recovery stroke, and these two are asymmetric, and this allows to break the symmetry and achieve net self-propulsion in the forwards direction. The two flagella mostly exhibit in-phase synchronous motion, but occasionally they go out of phase with a random occurrence. And macro swimmers like Chlamydomonas swim in helical trajectories, perform taxis, which is the directional response to a stimulus. And for instance, they can swim towards light, and they can swim towards food, against gravity, etc. And some of these organisms also have been known to predate on smaller microswimmers too. So one question that comes is, how is flagella beating related to the whole cell motion, and how is this controlled to perform these tasks? And our approach to address these questions was to develop a theoretical and numerical model that can be extended to a number n of flagella. And here I'm going to show it only for two for simplicity. So the flagella and the body in this model are represented by spherical rigid beads. And the flagella beads are bound to move in circular orbits, which are organized in a rigid scaffold. And the cell is moving, as I said, at 0 Reynolds numbers in our approximation, so inertia is negligible. The model is made fully three-dimensional by tilting the flagella orbits, and a tangential force is available, present on the orbits, and this induces net motion via the interactions between the beads mediated by the fluid. And this is obviously a very simplified mechanism compared to the one that allows real cells to swim, but nevertheless, it allows for the same swimming output. So with this model, we show that we can reproduce all the important features of the motion of biflagellates, including helical trajectories, phototaxis, etc. And there are two ways to power this motion, this model in a way. One is by imposing the flagellar forces, and one is by imposing the gait, by imposing the kinematics of these beads along the orbits. And the equations of the model, very briefly, can be obtained by imposing that Reynolds number is 0, and then balancing the driving force on each flagellum with the tangential drag force exerted by the fluid on the same flagellum, and that's equal to 0 because the inertia is negligible. And then we rewrite the drag as a function of the bead velocity, the fluid flow, and the friction term. If we add on top of this the torque-free and force-free condition, because remember, there's no external forces or torques on this system, we obtained a closed system of equations that can be solved numerically. And with this, we reproduce all the 3D features, for the first time, the 3D features of Chlamydomonas motion, including rolling motion around the main axis, superhelical trajectories, bit-controlled phototaxis. But here, I want to focus in the rest of my talk on something even more interesting than just the biflagellate case. And this occurs when we look at four-flagellated swimming organisms. When we have more than two flagella, obviously, things get more interesting because we have more patterns of gaits. And here we see 3 examples of microswimmers that have 4 flagella and similar morphological configurations, and they each have a preferred beat patterns for their flagella. So in terms of our model, that's here been extended to 4 flagella, we can describe the ones that have been observed in experiments, which are the pronk, the trot, with opposite pairs of flagella in antiphase, the gallop, where the phase difference between adjacent flagella is constant and equal to pi/2, and then the bound one, where pairs of adjacent flagella beat in antiphase. So these 4 have been observed, and each of the organisms that have been studied experimentally seem to prefer one gait, and sometimes, occasionally, can show a transition between 2 gaits. So what is the role of these gaits? Now, the obvious question is, how do they relate to the different tasks that the organism has to perform? And a group at Georgia Tech, in collaboration with Kirsty Wan at the LSI, tried to answer the question of which gait is the fastest, which one is the most efficient, effective to swim freely. And they realized a macroscopic robophysical model which reproduces the Reynolds numbers and swimming gaits of quadriflagellates. And this robot has a scale of a few centimeters. Its flagella are made by 2 rods and 3D printed joints, and powered by servo motors, and the microcontroller in the center is Wi-Fi operated. The comparison that was obtained when this robot was immersed in a high viscosity fluid and let move with different gaits, the comparisons show that the trot gait is the fastest in the robot, and this confirms what is observed in experimental live cells, which is, again, the trot is the fastest and pronk and gallop come next. In our model, in our system, though, let's see if I can get it, we simulate these equations and we have more freedom to explore different, oops, I've gone too far, apologies about that, a bit of lag with this video. We can simulate different fluid flows in 3D, and these are the projections, the side view and the top view. And here we see the trot gait, how it evolves in time in terms of the fluid motion, but also we can obviously compare the different gaits, and here we have the average speeds, and we confirm the results obtained for the live cells and the robots, with the trot being fastest gait. If we look at the in-beat dynamics, we see that the pronk, which is the synchronous gait, actually reaches highest speed during the power stroke, but then moves backwards significantly during the recovery stroke, whereas the trot moves steadily forwards. Finally, I wanted to quickly show you some other tasks and the implications that this model have on other tasks. So different gaits have affect the fluids in different ways, and this can have a series of behavior implications. For example, if we calculate the clearance rate, which is the flux across a disc placed in front of the cell, we can see that the cell in certain particular gaits has the ability of pulling, this is not working, pulling fluid towards its body. And in particular, the pronk gait, which is not the fastest, is the most effective of pulling fluids towards it, and so could be used for feeding. And finally, some gaits, like the gallop, clockwise gallop, and the bound gait, have got the advantage of disturbing the flow far away from the cell a lot less than the others. So this could be used for stealth mode swimming or quiet swimming, which can be used for predation or to avoid predators in the opposite case scenario. So in conclusion, flagellates display several distinct gaits and the ability to switch between gaits and control them in response to different cues. A minimal model can be used to reproduce the motion and study the difference between gaits. And our results suggest that the specific gaits might be used to perform specific tasks in consistently with evolutionary adaptation of these organisms. And this presents interesting potential for gait-based control of self-propelled artificial swimmers at low Reynolds numbers that don't require external fields to be operated. And with this, I would like to thank you for your attention, and you can find out more in the reference that are on the screen. Thank you.